Hey guys, so every single class stopped at a different point when it comes to the cell cycle. So I'm just going to start with interphase and go forward. If you want to jump to wherever your notes have stopped, feel free. Otherwise, I'm going to run through all of the steps here. So our cell cycle starts with interphase, moves into mitosis, and ends with cytokinesis. If you haven't looked through those slides yet, please do and let me know if you have any questions. But we're going to start here with a look at interphase. So interphase are steps where G1, or the gap 1 phase, where we're growing and producing proteins, the synthesis phase, where we're duplicating our chromosomes, and then the G2 or gap 2 phase, where we are going back to growing and making more proteins. So this is what it looks like in a plant cell and in an animal cell. And you can see that we have a lot of stuff going on in those center nucleuses. And then we also have the structure of the outside of our plant cell with the more blob-like form of our animal cell. So after we have done the preparation during interphase, we're ready to divide. And so we are going to go through what we call mitosis, which is essentially just cell division. One cell is going to become two identical daughter cells in this phase. And this is important because our mitosis section is where we're creating identical cells. There will be an instance where we don't create identical cells, so you need to make sure you know the difference. And the steps we're going to go through are our prophase, metaphase, anaphase, telophase, and then cytokinesis. This is a really great reference picture for you to use because it puts all of the steps side by side. So in prophase, this is called what I call the prep phase. So we have our chromosomes condensing. So you have your long strands of DNA that are part of each of your individual chromosomes, and they're going to condense down. And then you can see them in your cell as little X's, which we call our sister chromatids. Now you are going to have 46 of these X's in your body. That is not the case in these images because that's a lot. So please draw these images that you see into your notes. Our nuclear membrane disappears, which is why we have that dotted line around our chromosomes. The nucleus's job is to protect your DNA. And right now we need your DNA accessible so that we can split into two different cells. So the nuclear membrane is going to go away on this for just a little bit. And then our centrioles, or these kind of like blue earplug looking things, we have spindle fibers that come out of them. So our centrioles' main purpose is to help with cell division. And so right now they're growing what I consider like these creepy spider legs so that they can do that. So at the end of prophase, you can see we have our like clustered up chromosomes. Remember, these images don't show us the 3D, so while we are working to get those X's visible, you're not going to see them quite like X's right now. But those darker lines are going to be your chromosomes that are condensing. And then you can kind of see these little faint lines where there are spindle fibers growing out of our centrioles. In metaphase, this is the meet in the middle phase. So the spindle fibers are going to connect themselves to the centromere, or that like place where our sister chromatids are hugging. And they're basically going to try to grab the breadsticks. So your chromosomes are going to move to the middle of the cell and line up along the middle. And this is what's going to allow them to split nice and evenly. And this is why it's called the meet in the middle phase. So you can see our chromosomes are all lined up in the middle of each of our cells. And then the animal cell, it's really easy to see those spindle fibers coming off those centrioles. And you have them attaching to all 46 of our chromosomes so that we can split them apart, because right now we have duplicated DNA in our cells. Then anaphase, or the apart phase, this is where our sister chromatids are going to be pulled apart. So we're going to take our crisscross breadsticks and we're going to break them apart into individual chromosomes, because our chromatids with their little spider legs are going to go from the middle to start pulling them to opposite poles of the cell. So here you can see a really great image of it. And actually, if you Google mitosis under a microscope, this is really the phase that you can see the easiest. So we have our chunks of chromosomes being split apart and pulled to opposite ends. Telophase, I don't have like any cool way to remember this, so if you come up with one, let me know. But in telophase, we start protecting that DNA again. So our nuclear envelope comes back around each set of chromosomes. And then they start to look like chromatin again, which means that they're unwinding. So it's kind of like the slinky is becoming completely unwound. And you just got a tangle of stuff now. And then our spindle fibers, or your creepy spider legs, are going to break down and they'll actually dissolve back into the cell because they're no longer needed. Then we start cytokinesis. So PMAT, prophase, metaphase, anaphase, telophase. Those are the 4 that you really need to remember as the core of mitosis, and we'll see them again when we talk meiosis later on. So if you can come up with a way to remember PMAT, you'll be set. Then cytokinesis is technically the last phase of our cell division. So here's telophase. You can see a little bit of a circle forming back around all of our chromosomes, and those are new nuclear envelopes, nuclear membranes. In cytokinesis, this is where we have like everything's pulled off to different sides. Now we're actually going to break them apart into 2 individual cells. So in plant cells, because they have so much structure to them, we're actually taking a cell plate and forming it halfway. And so you've got your nucleus on each side of the cell, and then we start growing basically a wall in between. You can almost think of it like when a magician would like take the giant saws and like saw somebody in half. That's kind of what's happening here. We're creating a rigid barrier. But in animal cells, we form what we call a cleavage furrow, which basically just means that you've got this big blob. It's almost like taking a balloon and like twisting the balloon into 2 halves. That's basically what's happening. We're kind of pinching down in that middle to create 2 equal new cells. And in the end, they're going to split apart and you're going to see 2 identical body cells. So we have cytokinesis happening in animals, looking right here, so you can see how it's pinching in the middle. That's where our cleavage furrow is. And then in plants, we create this wall plate in between, and it starts forming up and becoming rigid. Now, how often do our cells divide? They all divide at different rates based on what our body is going to need. So your intestines might be replaced every 5 days, skin cells about every 2 weeks, blood cells every 4 months, liver cells every year. Remember, it's just based on growth and repair. So this is happening at all stages right now in your body in millions of different cells. And why do we divide? For growth and repair. We don't just grow our cells bigger. They stay small and they stay tiny. And that helps us out so that we can have things move in and out of our cells, but also if something happens to it, we aren't left with a gaping hole and you can't feel cells exploding on your skin. And now cells are not going to divide if we don't have enough nutrients for cell growth, or if we haven't replicated the DNA in the nucleus. And if the DNA is damaged, the cell will also decide it is not going to divide. And we're not going to watch that video. All right, so that is the end of our cell division notes. Please take those down and follow the instructions on Google Classroom to make sure you have your assignment done.